Dear friends, this topic we will study biosynthetic pathways of the secondary and primary metabolites in the plants. In this part 1 section, we will first see what is the difference between the primary and secondary metabolites and what are the different biosynthetic pathways which are the basic in nature for the plant, very important for the formation of different secondary metabolites. In this slide, you can see this is the general biosynthetic pathways for the secondary metabolites in the plants. There are two types of metabolites, primary metabolites and secondary. Here in this pathway, you can see what kind of primary metabolites are there and what kind of secondary metabolites are there. Here you can see three pathways are there. One is the formation of the triterpenoid type of molecules which are formed the of Mevalonic acid pathway. The other pathway is here is the acetate malonate pathway by which the formation of fatty acid occurs. Here the other pathway which is going to be used for the incorporation of the nitrogen atom is the Shikkick acid pathway. From this, the formation of nitrogen containing compounds are formed in the Shikkick acid pathway. Okay? The whole process starts from here after the photosynthesis, metabolism of starch, glucose, fructose, glycerin. So this basic metabolic pathway provides a skeleton how mevalonic acid pathway with the formation of terpenoids, how the acetyl coenzyme A and pyruvate play an important role in the formation of the fats, triterpenoids, terpenes, and as well as the amino acids in the plants. You can see the same biosynthetic pathway in a different manner. Okay? In the red blocks, you can see what kind of different amino acids are formed here. The glycolysis cycle gives the formation of pyruvic acid from 6-phosphate. This pyruvic acid then worked as a precursor for the formation of PAL type of amino acid, that is P for valine, A for alanine, L for leucine. Well, in the other part you can see the formation of DOX, deoxyxylenone, which with ethyl electrical phosphate with the formation of mevalonic acid. This we had seen in the previous slide also. Now you can see the phosphoenol pyruvate gives the formation of shikmic acid. This shikmic acid results in the three type of amino acid, that is phenyl, alanine, tyrosine and tryptophan, which are phenyl propanol negative and also called as the C6 C3 skeleton units, which are formed through this. The other thing we can see, the acetyl coenzyme A formed incorporates into the Krebs cycle, and the amino acid which we got is isoleucine, aspartic acid, methionine and L-lysine. Other pathway gives the formation of glutamic acid, L-arginine and L-ornithine. So this is the amino acid pathway which is occurred in the plants. In this shikmic acid pathway, you can see this is the general scheme which I had incorporated some of the enzymes also. You can see in this that the glycolysis pathway gives the phosphoenol pyruvic acid, that is PAP plus, that hydrochlose 4-phosphate PEP and hydrochlose 4-phosphate, that is coming from the pentose phosphate pathway. Then the aldol condition occur, and the coupling of these both two, it gives the DAHP deoxyarabinohaptosulfonic acid, which have a formation of carbocyclic intermediate due to the aldol reactions, and then the 3-hydroshikmic acid is formed. So why shikmic acid is formed? Sure, that is 3-dehydroquinate synthase make a formation of 3-dehydroquinic acid, and The 3-dehydroquinonic acid then form the 3-dehydroquinase to form the 3-dehydroshikmic acid. This 3-hydroxylic acid forms the shikmic acid formation. You can see three type of shikmic acids are there: 3-dehydroshikmic acid, shikmic acid, and shikmic acid 3-phosphate. Again, there is a phosphoenol pyruvic acid. That is, pyruvic acid is there. Again, it interact with the shikmic acid 3-phosphate to give the chorosmic acid and perfenic acid. This chorosmic acid gives the formation of tryptophan, an amino acid. चोरी कर अंत कर डाला, that is chorosmic acid and thrombotic acid, and then phosphoribosyl pyrophosphate to form the tryptophan. The part perfenic acid give the formation of phenyl pyruvic acid and phenylalanine, and the other one is tyrosine after the formation of pyruvic acid. So you can see in this part of shikimic acid what is happening. This is a 10-step reaction: 1, 2, 3, 5, 6, 7, 8, 9, and 10. Thus, the fifth step you can see there is a formation of shikmic acid. You can see the structure of shikmic acid seems like a joker ki topi. 1, 2, 3, joker ki topi ko fundna. Another eye in a creative way you can see, and आगे तितर पीछे तितर कुल तीन तितर. The formation of shikmic acid, there is a shikmic compound. There is also an shikmate compound. It means तितर के आगे तितर पीछे तितर कुल तीन तितर. This is shikmic acid pathway. Okay, some of the enzymes are. Now we can see the mevalonic acid pathway. Mevalonic acid pathway, again you can see I had told you that the pyruvates play an important role in acetyl coenzymes. Here you can see acetyl coenzyme A and acetyl coenzyme A. Both unit may form to form a HMG covalent with hydroxymethyl butyrate. This hydroxymethyl butyrate then form mevalonate. Here it is formed the mevalonate was presenting in this form. It is COOH, then mevalonic acid, which gives the formation of 5-phosphomevalonate, and the incorporation of ATP, it gives the pyrophosphomevalonate. So the incorporation of ATP gives an extra phosphate unit from this. So you can see titar ke aange titar, total 3 titar. This mevalonic acid pathway and the formation of mevalonate. Now the important part of this biosynthesis is the formation of this isopentenyl pyrophosphate. The formation of IPP. You can see 5 carbons: 1, 2, 3, 4, and 5. Isopentenyl phosphate. The isomer of isopentenyl phosphate is DMAP. You can see a positioning of this bond is just changed here in DMAP. So IPP and DMAP, C5, C5, it comes to form the G5, G10, C10, that is guanylyl pyrophosphate. Isopentenyl pyrophosphate, dimethyl allyl phosphate, both are combined to form a C10 carbon compound, that is guanylyl pyrophosphate. Again, isopentenyl pyrophosphate come to form 10 plus 5, 15, that is farnesyl pyrophosphate. So IPP, DMAPP, GPP, and then FPP is formed. This is mevalonic acid, which mevalonic acid plays an important role. You can see the same thing in different manner, that is mevalonic acid form and the methyl phosphate to form the IPP, and this IPP is for isomer of it is dimethyl allyl pyrophosphate to form the 10 C10 unit, C15 unit, C20 unit, C25. You can see the addition of IPP in at every place to increase the plus 5 unit to form the different type of terpenoidal compound, that is hemiterpene, iridoids, sesquiterpenes, sesterpenes, triterpenoids, steroids, tetraterpenes, and carotenoids. Okay, so the isoprene is the basic unit for here formation of this type of terpenoid compound, that is C5H8. You can see the rubber is formed by addition of head to tail unit of isoprene unit. Beta carotene is formed, the carotenoids. Triterpenes are formed like the acids, sesquiterpenes are formed, gibberine, a compound of officinal. And for the steroid, very important part of this is the formation of squalene and rate limiting step. So it's an important part here for the formation of steroid. Now we can see the other pathway that is called as DOX pathway, deoxycellulose pathway for the formation of compounds. The glucose formed with acetyl coenzyme A and acetoacetyl coenzyme A give the formation of mevalonic acid. This is the conventional method which you had seen. The other pathway it is formed is the formation of glyceraldehyde and pyruvate give the formation of TDP, that is thymine diphosphate participation to form DOX, deoxycellulose phosphate, and its DOX results in the formation of again isopentenyl diphosphate for the further reaction. So you can see the formation of IPP is important in the pathways, whatever it be, that is DOX or the simple formation of IPP from the glucose. Now this is the last pathway, that is the acetate malonate pathway. Why we had seen malonate? Because you can see the formation of malonic acid, malonate pathway. Okay? You can see the acetoacetyl coenzyme A with the carbon dioxide give the formation of this. You can see I had presented in this way acetate plus malonate, that is palmitic acid, stearic acid, oleic acid, linoleic acid, linoleic acid. These type of different fatty acids are formed in this pathway. Sequence of the formation of olefinic acid acid in the plants, and that is the formation by acetate pathway for the plants. Thank you. Happy learning.